Avrille Aiello, and I've spent this last year in the Hogranson lab in the Department of Cell Biology and Anatomy working on a very interesting project involving freshwater fish from Iceland called Arctic charr. Arctic charr is a single species of fish, but depending on where they live, they can actually look quite different. So, since there are multiple microhabitats within a larger lake, the unique environments that the fish experience have caused them to change their shape in order to be best suited to living in those conditions. As you can see, the exterior anatomy of these Arctic charr vary in shape, size, and color, and all of these alterations help them be successful in their unique microhabitats. My research on the environmental influence on body shape goes physically deeper to look at the skull bones of the Arctic charr. Generally, studying how a species adapts physically to new environments is difficult because changes in shape and size happen gradually and very slowly over several millions of years. However, this is not the case for Icelandic Arctic charr. They experienced very rapid changes in size and shape in response to new environments when they moved into Iceland as the glaciers retreated around 10,000 years ago. Arctic charr make an excellent micro-evolutionary model for studying adaptations to new environments because they went through this very dramatic changes in a relatively short amount of time. I'm examining the face and head shapes of Icelandic Arctic charr ecotypes that live in 3 very distinct environments of the same lake. The purpose of this study is to understand how various environmental factors such as diet, depth of water, sunlight, or even predator-prey relationships can impact different face structures. We also want to know how these various environmental needs are accommodated in the entire skull of the fish. We are using 3 distinct ecotypes of juvenile Icelandic Arctic charr to answer the following research questions. First, how do environmental selective pressures act on specific structures in the skull and on the whole skull as a complex? And second, what are the physical adaptations in the skull of each of the charr ecotypes? Around 20 specimens of each of the 3 Arctic charr ecotypes were shipped to the University of Calgary from Iceland to scan and analyze. All individuals were raised in captivity with the same diet and conditions for studying microevolutionary patterns of head shape. When the fish arrived, I fixed them in a preserving solution to prepare them for analysis. To start the data collection, I X-ray micro-CT scanned each fish using a protocol that I developed over the summer. The CT scanner will take cross-sectional images of the fish like these, which I can use to create 3D reconstructions of the charr. As you can see, we can create a very detailed picture of the internal anatomy of these fish, and we can gather precise shape data. These scans are really exciting to see because we're able to get 3-dimensional, comprehensive look at the bones just beneath the scales without dissecting the animal. After creating 3D models of the charr skulls, I can place 3D coordinates on the bones that I'm interested in through an automated machine learning pipeline. By placing these 3D coordinates, I can capture shape information from the structures. I must choose locations on the bones that are consistent across all the specimens so that we can compare these structures later in analysis. As you can imagine, even though these fish are the same age, they will not be exactly the same size. So to solely focus on the shape of the skull bones, I can use a program that will disregard all the other information not related to shape, such as differences in their size or position. With all of the 3D shape data that we gather, we analyze the differences among the 3 charr types at 2 levels. First, looking at the whole skull, and second, looking at the individual skull bones as a set of distinct structures. This allows us to determine whether the different environments have affected some parts of the head more than others, and how that influences the overall shape of the head in the fish. This research is still ongoing, however, the results will account for the environmental influence on the face shape of the fish. And so moving forward, this environmental information can help inform future studies on the genetic basis for physical adaptations to new environments. I learned every aspect of the data collection and analysis for this project from the ground up, and I've acquired a tremendous amount of practical research skills and techniques that have allowed me to accomplish all of these steps to the research project that I've outlined here for you today. I'm extremely grateful for the opportunity to study in the Hogranson lab this year, surrounded by extremely helpful and supportive lab members. I would also like to acknowledge all those listed on the screen for making this research opportunity possible. Thank you so much.